Right.
All right, I am starting the YouTube web stream. All right. Hi, and welcome in the next webinar in our Plantae webinar series. My name is Jason Padilla, and I am your technical host for today's webinar. Next slide. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all the ASPB members who are attending today. Your ASPB member dues support making this webinar possible. For any of you who have not yet joined ASPB, you can join today and use the discount code %10 to receive a 10% discount on your registration. ASPB members get early access to this webinar. You can learn more about ASPB and opportunities we provide at aspb.org. Next slide. If you haven't already, be sure to check out the Plantae YouTube channel. We've had so many amazing webinars, and this is just going to be added to a library of awesome webinars that we've had over the years. And so be sure to check them out. And if you're not already, again, be sure to follow us and check out our YouTube channel. Next slide. Before we get started, I'd like to go over a few housekeeping items to make sure that you get the most out of attending today's webinar. First, if you are experiencing technical difficulties or issues with Zoom, please let us know about those by emailing me at jpadilla@asp.org, or again, you can just message me directly in Zoom. If you have questions for our guest speakers, please add them on to the Zoom Q&A box. Our moderator will be sharing them with the speakers when it's time for Q&A, and we'll try our best to get those answered. And finally, if you're having trouble connecting or need to leave the webinar, know that this webinar is recorded and will be, again, made available on our YouTube channel. So again, be sure to follow our YouTube channel to receive notifications when the video recording is ready. And with all of that said, I'm going to turn the floor over to our facilitator, who will be our Plantae Fellows, and will be moderating today's webinar. With that, take it away, Andy and Rajarshi.
Hey, how's it going, folks? Welcome to the Plantae Presents Plant Science for Startups. Today, we've got 5 speakers for you here today. They're coming from all kinds of backgrounds, as well as all kinds of companies doing all kinds of fantastic things. We're going to start off our talk today with Dr. Maria Sorkin, an organism engineer at Ginkgo Bioworks, before following with Dr. Kathleen Heffron, CEO of Forte Protein. And then following will be Dr. Wesley Glenn presenting for Ayana Bio. Dr. Daniel Westcott, the head of protein and texture at Climax Foods, before we close out with Adrian Lopez Velarde, the co-founder of Deserto. So with that being the case, and without further ado, let me give a little bit longer introduction for Dr. Maria Sorkin, who will be starting us off here. So Maria Sorkin is an organism engineer at Ginkgo Bioworks and has previously done their graduate work looking at plant circadian rhythms and how they contribute to plant physiology. They'll be talking a little bit about their work at Ginkgo Bio, as well as Ginkgo Biowork as a whole. So, Dr. Maria Sorkin, I'll head it off to you.
Okay, great. Thank you so much, Andy. And also thanks for Rajarshi and Jason for organizing this webinar and inviting me to talk. Excited to tell everyone about a little bit what we're doing at Ginkgo. Okay, and I'll go ahead. Oh, Andy, can you stop sharing your screen so that I can share my slides?
Absolutely.
Okay. Okay, and you can see my slides?
Perfect.
Okay. Right. So again, I'm Maria Sorkin. I work at Ginkgo. I'm an organism engineer, which at Ginkgo just means scientist and R&D. So I work on developing our high throughput plant transformation and screening capabilities here. I also work as a project lead to develop new projects for our partners and communicate those to our internal and external groups. Okay, so let's say you might be in this audience with a great idea that you would like to see turn into a new biotech company. And you think, what if you could resurrect the smell of an extinct flower? So this middle image that you're seeing on your screen is actually an artist's rendering of this fossilized plant on the right-hand side that went extinct in the 1800s. It was native to Hawaii and it went extinct due to human activity. And you think, what if we could bring back the scent of this plant, maybe turn it into a fragrance for a perfume company? And I presume the market for this such product would be fancy nerds. And you have your idea, but you're going to need the following things. You're going to need people to do the work. You're going to need money to pay those people. You're going to need money to buy your lab space. You're going to need money to buy the equipment and consumables for your great idea. And you're going to have to work with limited resources and time for several years. And so this is the challenge that really founded the mission of Ginkgo Bioworks. So our mission is to make biology easier to engineer. And this is our dream to help accelerate startups and established companies alike to reach their R&D goals. So when we were founded in 2008, we wanted to tackle these problems of startup companies to basically create an easy button to allow you to not have to spend the time or money to build or hire your cell programming platform. And while Ginkgo, I would not say is a startup anymore. Again, we were founded in 2008 and we are publicly traded now on the New York Stock Exchange. We did start out as a small startup. So these are some fun pictures of our 5 founders. They were 4 graduate students from MIT and their advisor, Tom Knight. And you can see that at first we were hauling freezers out of MIT's basement, buying supplies off of eBay, manually building the lab that now today houses 300,000 square feet of our foundry here in Boston. So we remember what it was like to be a small startup that was strapped for cash and time. And so the way that we address these problems for new startups is with the following 3 assets that are the basic foundation for our company. So we have invested over $1 billion and over a decade of work to develop our platform here at Ginkgo. So this is based on flexible automation. So the video that you're seeing is from our foundry, which you can kind of think about as a factory for biology. This allows us to work at a scale that simply wouldn't be possible from the standpoint of an individual scientist. We've also collected a massive proprietary collection of metagenomic data that exceeds Uniprot's database by over 10-fold. This includes all of the DNA parts, different strains, cell lines that we can query for our customers' projects to find them the gene or biomolecular pathway that they're looking for. We've also recently partnered with Google Cloud in a $250 million collaboration to help AI speak the language of DNA. And so we're also entering into more AI-guided projects with our partners. So we've been able to successfully expand this platform to work for lots of different industries, from agriculture to pharma, from established companies like Syngenta, Bayer, Corteva, to younger companies such as Iona Bio, which I wanted to highlight here because you'll actually be hearing from Wesley Glenn, who is the VP of Innovation at Iona later in this talk. In terms of what Ginkgo can do for plant biology specifically, I wanted to highlight a few specific offerings. So in terms of trait discovery, we can use that massive database that I just mentioned to identify unique genes or mechanism of action, different biosynthetic pathways that are relevant to your trait of interest, whether that be herbicide tolerance, disease resistance, insect control. We're also the industry leaders of enzyme optimization through our protein engineering capabilities. And I also wanted to highlight our expansion into efficacy testing and screening. So we can now take those discovery libraries that we've identified through our large data sets and screen them in-house using plant cell screening systems. Also, additionally, not shown on this slide, but we recently established a strategic partnership between Ginkgo and Bayer's Ag Biologicals team. And with that brings their team's collective knowledge and expertise from years of successful R&D that brought products like Serenade, Sonata, Requiem Prime to market. And so we also have deep knowledge in that industry as well. So we've been able to successfully expand this platform to lots of different industries. For example, Microba is a human gut microbiome research company. Lygos makes bio-based materials for home care, water treatment, fragrance, and agronomy solutions. And I don't think I need to explain to this audience who Bayer is, but we do think that our platform has the capacity to accelerate R&D for small startups and established companies alike. And by the way, we're already expressing extinct plant aromatics and microbial hosts in partnership with the cosmetics company Archaea, so you can't use that idea. But for all you fancy nerds out there, you can now purchase scents such as Floating Forest, Solar Canopy, and Haunted Rose. And with that, I just want to highlight a few people from our business development, commercial, and marketing team. Brennan, Dia, and Josh. Their contact information is here on the slide if you want to reach out to them about working with Ginkgo or questions you might have about us. And I'm happy to answer any questions. Thank you very much.
Thank you very much, Maria. I'm going to go ahead and turn it over to Rajarshi, who will be fielding your questions for Maria. Go ahead, Rajarshi.
Yeah, thank you, Mary. It was an excellent talk, and you're actually doing a very, very interesting, innovative work in your field. And so there's one question from Sarvesh. It's asking, would Ginkgo collaborate with the lead developed countries like Nepal in plant tissue culture lab? Any investment opportunities in our country? So he's exactly asking that, are you open to collaborate with the other countries like Nepal?
Yeah, absolutely. We have collaborations with companies all over the world. So certainly we are happy to work with small companies. In fact, those are the companies that need us the most, we think. And so, yeah, certainly. Thank you for that question.
There's another question, that which type of plant enzymes do Ginkgo or its clients see as most in need for optimizing, and optimizing in what respect?
Yeah, that's a good question. I think I'll probably give a kind of vague answer here, which is that I think the primary applications for plant biotech, you think of disease resistance being a big one, herbicide tolerance. These are the types of traits that companies tend to be really interested in, that farmers tend to be very interested in. Yeah, I'm trying to see oh, okay. Optimizing in what respect? Usually, I mean, this could be identifying a new mechanism of action, or typically customers might come to us with an existing enzyme that works for, one set of targets, but they want to expand that target. So we can engineer an enzyme to make it more promiscuous or more specific, that sort of thing.
Okay, another short question. So it is more of a personal sort. So how did you go from your PhD or postdoc to working for Ginkgo? Does Ginkgo have lots of job for plant biologists? So this
Yeah,
is the question.
I love that question because yeah, I worked in a very basic plant biology lab and on a very basic plant biology project in grad school. I worked with Dimitri Nusinow at the Danforth Plant Science Center on circadian rhythms and Arabidopsis. So not very, I would say, industry friendly but in any case, I ended up here. And I decided to apply for both academic postdocs and industry positions, and really just wanted to try something new. And Ginkgo saw something in my presentation that allowed me to come work here. So I think, I could spend a lot of time answering that question, but I'll leave it at that.
Yeah, thank you, Mario. We can move to the next speaker. There's overwhelming questions for you, which you can, I think, use the chat box option to answer. And yeah, over to you, Andrew.
Thank you.
Andrew, I think you're muted.
Andrew,
Andrew, you're muted. Yeah.
Apologies, guys. Our next speaker will be Dr. Kathleen Heffron, a Cornell microbiologist and CEO of Forte Protein, a company that is focusing and specializing on the synthesis of animal proteins in plants. Go ahead and take it away, Kathleen.
Thank you. I'll just share. Okay. Just need to share my screen here. I think this is it. All right. See if I can get it to. There we go. Yeah, thank you very much for inviting me. Yes, my name's Kathleen. I am the founder of Forte Protein. I also teach microbiology at Cornell University. I have over 600 students, I think, this semester. It's really super crazy. But I decided to start this company based on a lot of research I've done over the last, many years, and Forte Protein is the result of it. So in Forte Protein, we can grow any protein in plants. We use a molecular farming technique, and I'll talk a little bit about what molecular farming is in general in this talk. First, the thing to tell you is that molecular farming started in its infancy, maybe 20 years ago. We were very interested in making proteins in plants using biotechnology, and these would be vaccine proteins. So we were able to show years ago that edible vaccines could in fact work, or producing proteins that were therapeutic in nature, such as monoclonal antibodies or other types of proteins. We could use biotech to produce these in plants, and it would work quite nicely. This just gives you some ideas of why anyone would want to do this in the first place. We don't necessarily need refrigeration, syringes, needles, trained medical workers to produce vaccines in plants. And this made things very accessible for folks who were living in low and middle income countries. So we were very interested in pursuing this as a field in general. This is just an example of when we've used plant-made vaccines, or in this case, this is monoclonal antibodies. We produced monoclonal antibodies in tobacco plants and actually used them in a previous outbreak in West Africa, where several Americans became infected with Ebola virus. And we were able to use this as an experimental treatment that seemed to work for them. So it just gives you an idea of what plant molecular farming could do. And I'd like to show this slide to provide an idea that this could make a really big impact on folks who live in low and middle income countries. Not only can we completely purify the vaccine or therapeutic protein from plants, as I'm showing on the top here, but on the bottom, we could also just use a crude extract. We don't have to necessarily purify, and we can provide this to folks living in places who have, diseases that, what I guess you call them orphan diseases. I also think we could provide just the technology and know-how to these countries, and folks could make their own vaccines to address their own, issues in their own countries. So from that, I decided to look at the prospect of making plant-made animal proteins using molecular farming, and I got into this field actually really after doing some travel through Cornell University, and getting an idea of, we really have, certainly in sub-Saharan Africa, a lot of people living there, a lot of people on the way who haven't been born yet, certainly not enough protein to go around. and sustainability is a global concern. We also have an issue of noticing that people in places like the States have have an increased interest in going plant-based. So this got me thinking about ways we could use molecular farming. We certainly have to change the way we're producing our food, how we're growing our protein. What is the carbon footprint of the animal protein we're producing right now? And so there's this is really giving you an idea of the whole alternative protein landscape that you may be aware of. And often we think of this as three columns, and I'd like to introduce molecular farming as a fourth column. So on the top left, you can see cultivated meat. This is it's not quite out there yet. Sometimes we hear about it. It's where we have cell cultures producing meat proteins. So it's quite expensive at the moment, and but it's up and coming. On the bottom left, you can see mixtures of plants. This might encode, an example of this might be Impossible Foods or Beyond Meat, where they're actually using plant tissues that have been processed in some way to resemble meat products. On the bottom right, we have precision fermentation. This is where we use microbes. So we might be engineering a microbe such as a yeast or a bacterial cell to produce actual animal proteins that we might, be able to use in novel food products. And then at the top right, we have molecular farming. And this is where we can produce the same proteins in plants. And plants have a lot of advantages, being low in greenhouse emissions and easy to grow and expand, this sort of thing. So this slide here just gives you a better feel for what I just told you. If you're imagining, let's say, having a glass of milk, on the left we have cow's milk. Then we have plant-based milk. This might be soy milk or pea milk. We've heard of that sort of thing. And then we have precision fermentation, where a company such as Perfect Day, I think, is making some dairy products. And these would be all produced without using animals at all, but using microbes in some way. And then finally, on the right, we have molecular farming. So it kind of resembles precision fermentation, but we're using plants. So it's kind of separate in its own way. And we're using a greenhouse instead of bioreactors. This gives you an idea of what kind of environmental impact we would have using molecular farming. And I'm showing you some simple examples here of a carbon footprint, how much carbon dioxide is released per kilogram of a product we're making here, such as cheese proteins or milk or eggs. And of course, something like red meat has a very high carbon footprint. It might be something like 27 kilograms of carbon dioxide released per kilogram of beef made. And then, of course, because we're using molecular farming here for Forte protein, we're just really having an equal or sustainable amount of carbon dioxide released. We're not really making any emission of carbon, or we might even be considered carbon neutral in certain instances, or carbon negative, I mean. So I think the future looks very promising for this field, and I'm excited to be in it. I'm giving you a couple of examples of other countries or companies, just so you're aware. Here is Moolec. Moolec is making it seems to be a heme protein, some kind of, might be myoglobin. This is soybeans, and so that's quite exciting. Here's another one. Nobel Foods is making casein and other dairy proteins in plants in such a way that they're going to end up with cheese, and it's all plant-based, but it's real cheese too. It's the real dairy product. And then we have Forte protein, and we're in a bit of a stealth mode, so I can't really tell you right now all the secret details about what we're doing, except to say that it's all very super cool, and just wait, you'll see it very soon. And that's all I was going to say. So we're housed at Cornell University. We're still in the incubator space there, and we're motoring along and having a lot of fun as we move. Thank you very much. I'll stop sharing.
Thank you so much for that fantastic talk. Once again, let me turn it over to Rajarshi here as he fields the questions that I'm sure are coming in at you.
Thank you very much for your talk, Kathleen. So there are a lot of questions for you coming up in the chat box, and one interesting question is: are there any commercial successes in plant-produced vaccines? And in case of crude plant protein extracts as therapeutics, how do you deal with the issue of correct antigen dose? Like, it's a bit of a technical question, but yeah.
Oh, sure. So yes, there are plant-made vaccines that have been commercially available. There's a number of companies right now where you can find these sorts of things going on. One that actually, I guess it just closed, but it was Medicago. They made a COVID vaccine. They closed for other reasons. I think it was a business strategy change. But there are a number of companies that I could list off for you if you're really interested. Often we use we may try to homogenize plant material before to provide the appropriate dose. So it wouldn't be a matter of going and eating a tomato and thinking that you're going to get appropriate immunization. You might have to do something like make it into a lyophilized powder and then resuspending into a tomato paste or juice to be certain of the concentration. So we do have to think about things like this. So that's something to mention.
Okay, another question is, what are the type of plants that you use for growing the animal protein scene?
Okay, so people are using all sorts of things. So I mentioned soy in one case. I think I know someone else who's using sunflower seeds and oil to produce some other proteins. A lot of folks use tobacco because it's just very easy. The FDA actually likes tobacco because it's a non-food crop, so they feel that it won't get mixed up in any kind of current food supply. People use lettuce, so there's all sorts of different types of crops. That's how I'd answer that.
Yeah. And what about the GMO issues which you currently face? Like, how do you circumvent it while producing these vaccines?
So, super great question. Some companies are struggling with that right now, and I'm hoping that in the future, near future, we'll see some advances in regulatory strategies in such a way that it's more not so cumbersome to go through those set of hurdles. But some of us, such as Forte Protein, don't make GMOs. We don't make transgenic plants. We use a transient system. It's part of our stealth mode right now. So I can't tell you too much, except it's not a transgenic plant. It's a lot easier for the FDA to handle. Thanks.
Okay, that's pretty much it. But we have many questions which I hope you can answer here. So yeah, thank you very much for your presentation. And over to you, Andrew, again.
Wonderful. Welcome back. Well, without further ado, we do have a third speaker. It's going to be Dr. Wesley Glenn, currently Vice President of Innovation at Ayana Bio, a company specializing in utilizing plant cell cultivations to increase the nutritional density of their health and wellness products. And it's a role that Wesley is certainly not unfamiliar with, having been working on plant metabolism and plant stress responses for his entire graduate and postgraduate career. Without further ado, I'll hand it off to you, Wesley.
Thank you. I need you to stop. Okay, there you go. Our diets are one of the biggest, if not the biggest contributors to our health. But unfortunately, many of us seem to be falling short. I won't bore you by reading out all these statistics, but let me just say that the outlook is pretty bleak. But central to our mission here at Ayana Bio, we think that botanicals can help remedy some of these shortfalls. But it turns out that making botanicals is really quite difficult. I represented Ayana Bio at the International Conference on the Science of Botanicals last year, and I can't tell you the number of people who I met who had to throw out their batches because they didn't have the right plant, or they threw them out because they were out of spec, meaning that they had high heavy metal concentrations or high levels of insecticides, for example. But we think that plant cell cultivation can remedy some of these issues because, after all, we get the best of both worlds. We get the fully controlled production of precision fermentation, and we get the phytocomplexity from whole plant agriculture. At Iona Bio, we use fully authenticated source materials from plants as well as defined feedstocks to create optimized plant cell line and medium pairings that go into fully controlled production and allow us to create rich phytocomplexes that are standardized and consistent every single time. So I'm speaking to a group of plant biologists, and I don't have to tell you all this, but plant cell cultivation is not new. In fact, the foundational technology was published in 1902, and it was used in earnest in academia starting around the 1950s. A couple of marquee examples that have hit the commercial space are using plant cell cultivation to produce paclitaxel, which is an anti-cancer agent normally isolated from the yew tree. And in the cosmetic space, there's an interesting example of a Swiss apple that didn't senesce for years. So scientists made a plant cell culture out of this particular plant and created an anti-wrinkle serum that you can put under your eyes. So you'll notice that these two examples really are in the high-end markets. They're in pharmaceuticals or in cosmetics. But what we want to do at Iona Bio is something different. We want to enter the food space. We want to make functional foods, functional beverages, dietary supplements, and even get into sports and nutrition. So our products look a little bit different. They're botanical powders. They have the full spectrum that you would find in a native tissue like a leaf or a fruit, for example. We can also make enhanced powders that have higher concentrations of specific bioactives. And because this is dried biomass, you can really manipulate it in similar ways as you can native tissue, and you can get extracts of molecule classes, or you can get down to single molecules in some cases. And the way we want to use these products is by putting them into consumer packaged goods, which are normally optimized for taste and flavor. We think that by adding our ingredients that we get from our plant cell cultivation platform, that we can create better for you products as well as better for earth products. A couple of examples I like to talk about are our Plant Cell Advantage Sage, which you could potentially put into something like tomato soup to increase the amount of antioxidants that you get. You could also take our Plant Cell Advantage Marigold that has high lutein concentrations and put it in something that's already very orange like cheese doodles and get higher concentrations of molecules that support eye health, as an example. And the way that we get to these ingredients is by using our platform that we've named Ascent, and I'll let you read what that stands for on the bottom left of your screen. We can take any source material. We can take something like a saffron flower or a blueberry or a cacao pod, or a cacao seed from a cacao pod, and put it onto a medium that contains phytohormones and create a callus from it. And from there, we can put this material into a suspension culture, make a suspension culture out of it, and just by putting it in liquid format. And from there, we can expose it to a variety of different growth conditions and a variety of different elicitors and elicitor concentrations that are a part of our media matrix. So the whole premise here in our media matrix is that we don't have to guess, we can test. We can figure out those conditions that allow us to have higher productivity. And the way that we do that is by going into screening mode. We can use colorimetric assays in some senses, and in other instances, we can use mass spec-based techniques. Then from there, we work with our partners, Ginkgo Bioworks, where we can look at transcriptomics to see which genes are expressed under specific conditions, or use metabolomics to see which molecules are present under different conditions. Each one of these data sets is useful and insightful in its own right, but we can also combine or correlate these data sets in what I call combinatorial analytics to get even more and even deeper insights into what's going on within the cells. Then from there, we can pare down conditions and start to revise either the cell line itself or revise the medium that it's growing in. And then finally, we do a little validation of productivity. And what I'm not showing on the right side of the screen is that we end up with a cell line and medium pairing that we can use for years and years that produces our molecules or molecule of interest. And I can't emphasize our relationship with Ginkgo Bioworks enough. You heard from Maria Sorkin earlier. She's one of the scientists who I have the pleasure of working with at Ginkgo Bioworks, as a fantastic scientist, a great person to brainstorm ideas with and think about how to get even more conceptual insights from our data. I don't know if Mike Torrance Spence was able to hop on the phone today, but he's also another plant biologist who is a fantastic resource as well at Ginkgo Bioworks. Just to give you a teaser of some of the data we get data spanning the entire central dogma of molecular biology from epigenomics all the way down to metabolomics. And of course, we look at the phenotype and how it relates to some of these multi-omics data and are performing phenomics in that way. I don't know if any of the scientists on my team are on the call today, but Michelle McKee is leading that effort. Fantastic scientist, and we're getting many, many different insights from doing this multi-omics approach that's also interoperative. And finally, we're able to control and understand how we're producing the molecule or molecules of interest, and importantly, understand what disables that production so that we hit our specification every single time. And with that, I'll just say follow us on social media, on X or LinkedIn to see the latest happenings. And I'll put my contact information up here so you can email me at any time if I don't get to your question today. And I'll just say thank you.
Perfect. Thank you so much for that, Wesley. As previous, please do enter your questions that you may have on the question and answer box, and where Josh will go ahead and take it away from there.
Hello, Wes. Yeah, thank you. It's a very inspiring presentation. And yeah, so I want to ask a very personal question. Like after doing the PhD and postdoc, like how you can motivate us, the students of plant science, to actually do the thing which interests us, rather than academics. Like how to get into this field and what motivated you actually?
I was very motivated by doing science that had a translational component. So I would say that my PhD and postdoc were more basic science questions that I was looking to address. But I found that biotech was the spot really to find a translational element to it. And I think that many biotech companies, they don't need you to have done something with a translational element during your degree, but they want to see how well you think. They want to see how you can approach problems in a creative and innovative way.
Okay, that's great. So one question is that what is the most demanded product which according to you, like in this startup, like what is it most from the consumer point of view?
Yeah, so as I mentioned, plant cell cultivation has been around for many, many years, and people have used it already for pharmaceuticals and cosmetics, but not as much in the food space. So it's pretty new. But what I can tell so far in preliminary talks is that people are very interested in going after hard to access plants such as saffron or rhodiola and making botanical ingredients from there in a really consistent format. So something like saffron, I would say, is something that people have been very interested in. And we just made a press release with that just a couple of weeks ago.
Okay, so are you also interested in this in the medicinal plants or the underutilized plants, which actually like people are not using it as such, but yeah, they have the huge potential, especially the wild type varieties. So what's your approach on that?
Yes, we are interested in all sorts of plants. I would say that our primary focus today is looking at food-based applications that can improve health outcomes. That being said, there are plants like saffron who are understudied, plants like saffron that are understudied precisely because there's not enough biomass out there to study them. So I'm optimistic that by having a robust platform, we'll be able to produce that biomass and ultimately fuel some of the preclinical, clinical, and food-based studies that will elucidate some of those mechanisms and potentially allow for even greater drug discovery.
Yeah, the another question which is popping out for everyone is, do you have any internship opportunities there?
Yeah, so we do not have any internship opportunities coming up this particular summer because we're in somewhat of a transition period, but we are looking potentially to do that in the following summer. So please keep a lookout on social media for that. And also shoot me an email as well. I can give you a more personal update.
Okay, thank you, Wes. Yeah, so we can move to the next speaker now. Yeah, so I want to introduce our next speaker, who is Dr. Daniel Westcott. He's the head of protein and texture at the Climax Foods, and the company, and he's already holding a doctorate in plant and microbial biology from UC Berkeley. And as he already informed, his work was on photosynthesis. And his professional journey encompasses roles as a farmer, cook, and educator. So all these skills makes him a very suitable man for this position. And Climax Food, I hope you have heard about it, is rapidly scaling to address the urgent need while preserving the culturally rich experience of delicious food. And so, yeah, over to you, Daniel.
Hi, thank you very much, Rajarshi, and thanks to Andy and Jason for inviting me, and also to the other panelists. It's actually really interesting to see the approach that everyone is taking here on what I think are really critical issues. So, are we seeing the right thing or my email? Let's see. We practiced, I promise.
We did. We did.
We did.
I can guarantee the success of our practice session. So
Good.
wonderful. It's working now. Just into presenter view and we'll be good.
Okay, cool. Great. Yeah, so at Climax Foods, we're interested in replacing animal dairy, right, entirely. And the approach to doing that means that it has to be cost competitive, texturally, flavor has to be the same, and nutritionally it needs to be dense. So cheese is a very, very nutritionally dense food, and so we can't ignore that at the outset and try and use inferior ingredients in order to make the same thing, because people will not buy it. You've probably experienced vegan cheeses and they are underwhelming in almost all cases. So this was the task we set out for ourselves. And the way that the conceit that we arrived at was that there was a lot of people working to recreate casein, the major protein dairy, from fermentation or to try and make an exact molecular copy of this protein. We started from the premise that maybe there's functionality in plant proteins that you can derive or understand and combine in order to get the same functionality, nutritionally dense and pleasing materials from available and scalable ingredients, rather than needing to solve the scale problem of a new technology. So I'm going to talk to you about what we call our ingredient discovery work. We have sort of two major arms. One is formulation intelligence, where we work on the data science of combinatorial ingredients. And then we have one where we're trying to develop new ingredients to enhance that space. So we started, like, there's a lot of cheeses out there, but they all come from a single protein source, which is casein, and it's a very unusual protein. It makes it somewhat difficult to just use through precision fermentation or other approaches, because that casein is an intrinsically disordered protein. It comes in 4 different kind of varieties. They're smushed together somewhat randomly. There's not a lot understood about the structure. And they're also assembled in vivo with an important fat globular membrane that gives the casein enhanced functionality. So it's not a trivial expression problem just to say, we're going to make a protein here and then make a cheese out of it, because the next application step becomes somewhat difficult. But there's a lot of effort to do it, right? So I put down a whole bunch of companies here who are working on this effort. And I think that technology is going to be excellent and an important part of our future food system, but it's just not there yet. So we're able to work on ingredients that are available. We can order them or grind them out of seeds, or go, potentially seek through sequences, more interesting functionalities. And we found some, right? So this is an example. What my colleague Avinash is holding down there is an example of one of the proteins that we functionalized from a plant, right? Taking a seed, extracting proteins, and treating them in such a way that those proteins can have a stretch and melt that we're interested in actually seeing in our products, right? On the left side is another approach. Like, since we know fats are important in these, we have developed a way to optimize fat mixtures, again, from available ingredients, using data science and using ML approaches to do Bayesian optimization to try and get those fats to perform in a much more believable way when you put them in your mouth. On the right side is just an example that we throw a lot of work into this, right? So it's an iterative process. We capture as much as we possibly can about qualitative and quantitative measurements as we make new formulations, and we try and then reduce our distance from dairy using that approach by driving directionality into our work through statistics and optimization. But So ingredients are really, really complex. And so what do? We try and run them all through. There's two regimes here. We can run in a high throughput regime and a low throughput regime. And one gives you sort of a broad view in your high throughput, but you also need a dense view in order to connect and create connections between ingredients and understand sort of the science there. So we really like dig into what a protein can do. We extract it. We try and understand water binding capacity, whether or not it's soluble, can it foam, what happens when you gel it, how much chemical activity might it have through its surface hydrophobicity and thiol content. So we're really like trying to get a comprehensive view of individual proteins and then expand that by using sequence similarities and high throughput approaches to sort of get a more broad understanding of what's out there. And then again, so we're interested in the other components of things. So fats, you can basically pick up a coconut oil and put it in your hand and it will melt. The temperature at which it melts is way too soon when it comes to your mouth and body temperature, and it doesn't give you a convincing feel. So what I'm showing you here on the right is this curve that dips really far. This is differential scanning calorimetry data, where we're actually measuring thermal transitions. That one's coconut milk. So at about 25, 23C, coconut fat is all the way done melting. And the other curve, the yellow one, or sorry, orange, is ghee. And that's like a critical regime of temperature that we're not seeing in coconut milk. So if you put it, or coconut fat, and if you put that in your mouth, you miss like the textural sensation. So we started by throwing lots of fats together and trying to understand how they crystallize and measuring them both through DSC and textural and sensory with assays to try and put together a more convincing fat. So we put proteins together from the plants and fats together from the plants in ways that enhance that functionality just using existing ingredients. And at the end of the day, we get a mozzarella, right? So how do we actually apply this? We put this in context of what's $1 billion market out there for mozzarella. But, of course, that's aspirational, sort of. We've learned that for us, we're not great at actually making products. We are much better at making formulas. So our current approach is working with big partners, and we've got a partnership with Bell to reformulate their vegan Baby Bells. And we're doing this sort of formulation intelligence approach using different ingredients in order to give a more convincing texture and mouthfeel and nutrition to their current product, which is basically starch and fat. And there's no protein and no nutrition in it at all. So our mandate is, from Bell, give us something with 10% protein that is as good as what we have on the market. And we're like, I don't know, about halfway through that collaboration and expect to see that on the market early in 2025. Briefly, though, we also are interested in fermentation, but not precision fermentation. We're interested in how cheeses are actually made. We want to retain the rich cultural heritage of foods because that is another adoption barrier to the consumer. It says, look, we've got all this great cheeses out here, and this one is the one I like from my, from my tradition or what my mom used to cook. And if you then ignore that and ignore that this actually has a rich history, you will not get the same kind of broad scale adoption. So we think that when you do fermentation and you adopt more traditional methods, you actually do two things. You make a more convincing flavor profile because metabolism is really hard to map individual molecules. You want potentially the microbes to do a lot more work for you, and you may not be capturing all of the molecules that make a really huge impact to sensory perceptions. And you do two things. You make a cheese that actually looks and feels and smells more like what people are familiar with. And there's tons more I can say. We've been doing this for about 3.5 years or so. So I've had a lot of experience coming straight from my PhD into a startup, and it's been an interesting ride. I've had to learn a lot of different skills to apply here, but I actually felt like I was pretty well trained to adopt these things after coming through a PhD. So retain that generalist perspective of things, even if you get specialized. And that's all I've got for presentation.
Yeah, thank you, Daniel. That was great. I will now pass on to Andrew for handling the question and answer for you.
Wonderful. Well, I guess I've got one for you.
Sure.
So oftentimes when we're thinking about, the kind of holy grails of plant-based meat or alternative products, we are thinking of dairy, we are thinking of cheeses. And it seems like you all have gone a long way towards kind of replicating a cheese-like mouthfeel. Are there any other popular, highly desired animal-based protein products that you all find that you are looking to also develop in a similar manner?
So given that there's like hundreds and hundreds of different types of cheeses. We got a lot of work ahead of us on that front. And so I don't think that we're reaching out into any sort of other animal-based products at the moment. Although mission-wise, right, dairy is only a small set of the climate impact of animal agriculture, right? So if we were to try and expand that mission, we would definitely go into things that would have, I mean, I think we have of the what, 23% global greenhouse gas emissions from the animal agricultural sector. There's maybe 16% of that is from dairy. So we would definitely pick the next slice that we could get to and actually approach. I'm always interested in egg because egg seems like a simpler problem, but everything seems like a simpler problem until you dig into the details.
That absolutely is the truth too. We have a second question for you here. Since your PhD was in photosynthesis, how did you decide to explore this field, which is so different, and how did you come up with ideas?
Yeah, it's not actually that different. By which I mean, like, yes, photosynthesis is a very specialized approach and understanding of but you have to understand, molecular biology, genetics, biophysics to some extent, chemistry, and all in the context of an organism. So when it came to applying sort of that perspective to physical problems, like we have to build things and make things, well, like and I'm actually looking at molecular sequences and interested in surface hydrophobicity. So all of those biochemical skills and trainings came into play, and the genetics came into play because we're interested in the diversity of the plant world in order to build these. So while the specifics of photosynthesis was not applicable, I cultured algae, right? Like, so I have an understanding of like microbiology and what we're doing for fermentation wise. There was just a lot of generalist and applicable skills that translated much better than I expected.
Wonderful to hear. And we'll hit you with one final short question here. As Climax is modifying proteins, what are the food safety considerations for these new proteins?
Yeah, that's a really good question. We don't do anything that's not food safe, right? So we don't pull any reactions out of thin air and do novel chemistry. We're using things that are already on the market, and generally we haven't found the need to do much modification. The real work is that we extract and isolate proteins more gently than is typically done in industry.
Fantastic. Thank you so much for answering those questions. We do have a couple more in the chat as well. We highly encourage you guys to keep asking those questions as our speakers will keep answering. So let me go ahead and turn it back over to Rajarshi, introducing our final speaker.
Andrew, you can share the slide. Yeah, hello. So we are coming to the end of the webinar with our final speaker, who is Adrian. Adrian is a visionary entrepreneur, and he has a year-long cultural exchange in Taiwan, and he actually co-founded the company which is now making leather from cactus, and it is a globally recognized brand. And now Adrian's latest endeavors also involves the launching of limited edition dessert variant crafted from agave. So yeah, these are all eco-friendly alternatives. And yeah, over to you, Adrian. You can now take over the stage.
Thank you very much for the warm presentation. So hello everyone, my name is Adrián López Velarde, and I am the co-founder of Deserto. Deserto is a plant-based alternative to leather featuring cactus, and we crafted this solution with the objective of helping the fashion industry and the automotive industry to reduce their environmental impact. Our mission, therefore, is to be at the forefront of innovation, offering high quality but sustainable materials. Sometimes there is a conception when we talk about sustainable materials that they might not perform at the level where synthetics or conventional materials do. So Deserto is a great example of how we can work with nature rather than against it in order to deliver materials that perform to the standards. And our vision is that our solutions can help to drive the sustainability transition for all these markets that we play with. Here we can see at the two main materials that we have developed, Deserto for the fashion industry and some of the brands that we collaborate with, and Desertex, which is our automotive dedicated material, and some other brands that we have worked with. Always featuring cactus for sure. and some of the awards and recognitions that are obtained not only for the material itself, but also for the agricultural practices that we implement. Sometimes when we pay too much attention into the material, we forget where the raw materials are coming from and the environmental impact starting from agriculture. Therefore, we have made sure that our sources are coming from organic plantations, free of agrochemicals. There is even no irrigation, given the resilience of cactus. So when we decided to use the Opuntia ficus indica species into our raw material program, being Mexican company, we thought that we should use a native species that is very resilient. Therefore, we don't have to reinvent the wheel and work with nature. And so this is what we have done. Our structure is in Mexico and also in Italy in terms of production. Until today, all of our cactus is still coming from Mexico, given the ideal conditions for cactus to grow in a very fast and resilient way with the least input of energy, agrochemicals, and water. And so you may be wondering why it's relevant to talk about plants in the as an alternative to leather or synthetics like polyurethanes or PVCs. And it is very important because the environmental impact coming from leather industry, it's very, very big. Not only is relevant to the tanning processes, which use a lot of noxious chemicals, but also from the environmental impact at the face of the growth of the animal, including deforestation. So how do we make our materials in a nutshell? So we rely on organic farming and harvesting techniques. This Opuntia ficus indica, it's endemic from central northern part of Mexico, mainly in the state of Zacatecas, Aguascalientes, and San Luis Potosí, where we source our cacti. And well, as we know, it belongs to CAM class species. It's much more efficient than C3 and C4 plants in terms of growing biomass with the least input of water. As a rule of thumb, you only need 200 liters of water to grow 1 kilogram of CAM biomass, compared to 1,000 liters of water needed to grow 1 kilogram of C3, C4 class species. So this gives us a very big advantage in terms of water savings. Then the drying of the mesh of the fresh cacti is done naturally on a solarium, so we don't rely on additional energy in ovens, for example. And then we do a separation of fibers, extraction of protein, cellulose, and hemicellulose, which serve a very important role in the mixing of our materials as a substitute of fossil fuel raw materials, and then creating the final product, in which in many cases is accompanied with a substrate, like a fabric or a textile, that gives the material mechanical properties in terms of tearing, tensile, elongation, strengths. So our main focus is sustainability, but also performance and making the material aesthetic. So we can customize the colors with organic pigments, and also working with designers to make the hand feel very appealable for the end consumer, and get the best chances for success while marketing this product. We also have performed a life cycle assessment in which you can have access to the data following the ISO 14004 and 14044 guidelines, so you can compare the impact of Deserto, animal leather, and polyurethane in the main relevant categories, which is eutrophication impact, cumulative energy demand, greenhouse gas carbon, water use, and carbon from waste incineration approach per square meter. Also figures apply for kilogram in a different summary, but this gives a lot of transparency and understanding for our customers when we try to push sustainable materials, so they can use this and inform the end consumer of the impact, the positive impact, or the savings that their choice is doing. Pretty much. Footwear to garments, handbags, upholstery, and much more. Some images, and I guess I'm running short in time, so I would like to stop here. And if there is any question, happy to answer. Thank you.
Thank you, Adrian. I will request Andrew to take the questions.
Yeah, we've got a few questions here in the chat. One of the first ones, and maybe one of the most topical, can other plants be used to make leather outside of cactus?
We always try to research cacti in general because it's the most abundant, sustainable plant that we have in Mexico. Of course, there are a lot of species, so we experiment with a few dozens of species, but we decided to work with Opuntia ficus indica, given the efficiency it has, because also when we were crafting the development of the material, we wanted to have a raw material that is scalable.
And so speaking of that answer too, when you're looking for characteristics of particular species or particular cactus that you can utilize for these downstream purposes, what kind of characteristics are you typically looking for in these species?
We're looking for a species that requires the least input of water, the least input of agrochemicals, that can grow in land that is not used for other agricultural systems that give us food. So we try to avoid cross-industries conflicts, and also that it grows in an abundant way.
Speaking of growth, there are other climates as well that are very similar to both Mexico and Italy. Are there any plans to expand the development of this cactus leather to other locations?
Indeed, we are currently in talks with the regional government in Sicily to explore the possibilities of relying on them for the supply of our Italian production line, because cacti also, the Opuntia ficus indica, grows very well in the Mediterranean.
And one last question here, maybe to kind of broaden our horizons here beyond just the cacti. What are the features of cactus leather that they might have that make them beneficial over, say, mushrooms?
The benefit is that the material has a very low impact on the environment in terms of energy, water use. We don't need to control a lot of parameters through the elaboration of the raw materials or the productions. So this minimizes the utility of energy. Also, it's a very dynamic material that can meet the demands of the designers for many different type of products. And finally, I will say that is the durability, which is very important for us to make very durable materials to ensure that the carbon that we are capturing inside of our products can be stored as long as possible.
Wonderful. Thank you so much for those answers. And of course, there are more questions in the chat. Please do continue to ask questions for our speakers here, as they will continue to answer. But in the meantime, let me go ahead and turn it back over to Rajarshi as he supplies us with our final section.
Thank you.
Yeah, so indeed it was a great experience learning from you guys. So as we conclude this enlightening journey to the vast landscape of plant science, we extend our gratitude to our diverse panel of speakers representing the forefront of innovation in plant-based industries, from the pioneers in plant-based food companies to the trailblazers in molecular farming, biotechnology, and the fascinating realm of cactus-based leather. Each speaker has woven a narrative of inspiration and possibility. So in a world where the intersection of plant science and entrepreneurship is creating waves of change, we hope this webinar has ignited your passion and provided practical insights for your entrepreneurial endeavors. So let the shared knowledge fuel your aspirations, and may you embark on your own transformative journey within the dynamic and sustainable future of the plant-based technology. So yeah, I will hand over to Jason.
All right. Well, thank you everyone for tuning in this webinar for our Plantae webinar series. We really encourage you to tune into our previously recorded webinar, and really, if you wanted to rewatch and reshare what you've learned from today's session, they will be available. And really, a special shout out to our moderators, and especially to all our speakers who've shared really insightful kind of innovations and really exciting things in the world of plant science for startups. With that said, we hope that we see you in the next session for our webinar, and I hope you all have a wonderful weekend. See you guys soon. Bye.